Hello and welcome to the final installment of the bacteria unit, and today we're going to talk about some genetic switches. They're called operons that function in the bacterial genome, and what they are is they're a single on-off switch that controls a whole cluster of functionally related genes. So one that we'll look at is for the production of lactase, and to make the enzyme lactase there's several proteins that have to be complexed together, and so what the bacteria genome does is it strings those genes together and has them function by one common switch. And so we're going to look at two types of operons. We're going to look at a repressible operon and an inducible operon. So again, these are a single on-off switch that controls whole clusters of functionally related genes. These genes are preceded by a promoter region that the RNA polymerase will recognize. The switch itself is called an operator. And the operator is the on-off switch. It's usually positioned within the promoter region or right afterwards. And the first one that we're going to look at for examples is a repressible operon. And so there's got to be a repressor. And this is a protein that can turn the operon off. It binds to the operator and can block the attachment of RNA polymerase so that the synthesis of mRNA cannot happen. It's a product of the regulatory gene located outside of the operon, and usually it acts as an allosteric protein, so there's a feedback mechanism, so that in the case of the tryptophan operon, it's only active in the presence of tryptophan, which serves as a co-repressor, because if there are high levels of tryptophan in the body, we don't, or the bacteria, we don't need more tryptophan produced, so it can act as a feedback mechanism as is expressed here. So if there's a metabolic pathway, and in this case there's 5 different enzymes that will be produced by 5 different genes that go into the production of tryptophan. Now, if the levels of tryptophan are high, there is negative feedback on one side by the regulation of enzyme activity, where the tryptophan can go back here, in the case enzyme 1, and shut it down so that no more tryptophan is produced. It can also act on the regulation of gene expression, so it can go back and function or inhibit the copying of all 5 or any one of these genes, and again stop the production of tryptophan unless it is needed. So, as I said before, the tryptophan operon or TRIP operon is a repressible operon, so it is switched on until needed to be turned off. So the bacteria will always be making tryptophan until we've got enough. So if we look at the diagram here, we've got the DNA that will be transcribed to RNA, which will be translated to a protein. Now, if tryptophan is present, it acts as a co-repressor. It will alter the shape of the repressor. The repressor will then bind to this area of the promoter region, and RNA polymerase cannot bind. So if tryptophan is in high levels, production of it is stopped until the levels go back down. So that's why it's repressible. It's always on until it's turned off. So again, here is the full thing of the TRIP operon. So we've got the DNA, we've got the promoter region, we've got the operator, and then we've got the 5 genes that we need transcribed and then translated for the synthesis of tryptophan. And as long as there are low levels of tryptophan, the repressor is inactive, the operator will be open, and RNA polymerase will transcribe the mRNA, which will allow for the production of tryptophan. Now, if tryptophan levels go up, the protein repressor, or the repressor, will become active. It will bind to the operator, and RNA polymerase will be blocked, and we won't make any more tryptophan until it's needed. So, real quickly, here is a video that will explain how that all works.
In the bacterium Escherichia coli, a group of 5 genes code for enzymes required to synthesize the amino acid tryptophan. All 5 genes are transcribed together as a unit called an operon. An operon is a group of genes that is under the control of a single operator site. A regulatory protein called a repressor can bind to the operator site and prevent transcription. When tryptophan is lacking in the environment, the repressor is inactive. RNA polymerase binds to the promoter site and then proceeds down the DNA, transcribing the genes for the tryptophan biosynthesis enzymes. When tryptophan is present in the environment, the organism no longer needs to make tryptophan. tryptophan binds to the repressor and activates it. The activated repressor now binds to the operator located within the tryptophan promoter and blocks transcription. The tryptophan repressor is a helix-turn-helix regulatory protein. When tryptophan is absent from the environment, the repressor is in an inactive conformation and cannot bind to the DNA to prevent transcription. When tryptophan is abundant, two molecules of tryptophan bind to the repressor. This alters the orientation of the helix-turn-helix motifs in the repressor and causes their recognition helices to fit into adjacent major grooves of the DNA. Thus, the synthesis of tryptophan occurs when it is needed, but is repressed when tryptophan is available.
So that's an example of a repressible operon. Now let's look at the opposite. So the opposite is what's called an inducible operon, and this one is only turned on when needed. So it's off until it's needed, then it's turned on, and the lac operon or the lactase operon is an example of that. So in this case, the repressor is always bound to the operator unless we need it. So in the case of lactase, when would we need it? Only when lactose is present. So in the example shown here, lactose is absent, so the repressor is active and no transcription takes place. If lactose is present, now it acts as an inducer. So in this case, a form of it called allolactose will bind to the repressor. It inactivates the repressor. The operator is then free to have the RNA polymerase bind, and mRNA is transcribed, and the proteins are translated into lactase. So as long as lactose is present, we can produce that. Now, even if lactose is present, in some cases, we're not going to produce lactase because glucose is a more efficient form of energy for that. So here's the case where lactose is present and glucose is scarce. Now, when glucose is scarce, there are high levels of cyclic AMP or CAMP, and what that does is the CAMP will bind to CRP, which is a CAMP receptor protein. That acts and helps assimilate the RNA polymerase so that the lactase can produce. So, if lactose is present and glucose is scarce, we get production of lactase. If lactose is present but glucose is also present, the CRP does not get activated. It does not bind to the CRP binding site, and the RNA polymerase won't bind as readily to that, and lactase is not produced, so the glucose is used up first or preferentially. And so the lac operon is only turned on when lactose levels are high and glucose levels are low. But again, it's an inducible operon, so that it's off until we need it turned on.
The lac repressor protein encoded by the I gene is expressed in the absence or presence of lactose. In the absence of lactose, the lac repressor binds to the lac operator site. Repressor binding to the operator blocks progression of RNA polymerase, like a DNA roadblock. Since RNA polymerase is unable to transcribe the lac structural genes, the corresponding proteins are not made. When lactose is present in the cell medium, it binds to the allosteric site of the lac repressor. This changes the conformation of the repressor. In this conformation, the repressor can no longer bind to the lac operator site. Without the repressor blocking its way, RNA polymerase is able to transcribe the structural genes. Thus, in the presence of lactose, the lac structural genes are expressed. The proteins encoded by the Z and Y genes are required for the metabolism of lactose.
So hopefully this makes sense now, and you can tell the difference between a repressible operon and an inducible operon. And again, thank you for your time.